Good morning. It's our Thursday morning, 10 o'clock live online review session. And this week, what we're doing, we're completing our exam 3 and our practical 3. So without a further ado, let me just share my screen for this week's learning module with you. And I'm gonna jump to the student view so it looks a little bit more familiar to what you probably will see. And if you scroll all the way down to what I believe is week 12, what you will see that for this week, I'm asking you to complete the lab practical 3 and the exam 3. So simply 2 elements of assignments that I'm asking you to do. And as always, I have prepared for us for preparation for the exam a study guide. And this study guide is available for you on your learning modules area at the Canvas. However, as you remember, the study guide includes gaps here and there. Often when it's a gap, it tells you that it's an important term or an important concept. So I really want you to pay attention to that thing. So that's why it's not a fully completed guide. And what we're going to do today on this morning's review session is that we're going to take our time and work our way through the study guide, filling in the gaps here and there, and just kind of reminding ourselves of the material that's being covered in this exam. You might also remember that one of the things that I do for our exams is that the exam questions are pulled from this study guide. So it's definitely worth of taking the time, working through it, and if anything seems unfamiliar or concepts that you're not totally comfortable with, it is a good time to revise those. And just in the name of being transparent, a quick reminder that I do not share the completed study guide with all the answers to the gaps anywhere else. So it's only covered during these review sessions, whether it's in person or whether it's online, but I will not be posting the completed study guide. It is something that only place where you really get it is by showing up, simply because of the fact that so much of the exam relies on the study guide. Otherwise, it really wouldn't leave you much to do. So make sure that you complete the exam 3 and make sure that you complete the practical 3. In terms of the practical, another thing that I wanted to share is a reminder that there's also a practice version of the practical available for you on this week's learning module. I'm not expecting you to complete the practice lab practical. It is an optional tool that's there for you to do. Many students do find it useful when they're preparing to take the actual lab practical that carries points towards your final grade. The practical lab practical practice version, the questions for that one are pulled from a larger pool. So often I encourage students to work through it few times, so you get a chance to see as many of those different questions that might come up, because you'll get a different set of questions every time that you work through the practice practical. So I encourage you to rely on that. That might be a useful thing to revise. When you're preparing for the lab practical, remember that we covered 3 chapters this week for this exam. So we covered the respiratory system, so there's going to be anatomy in there. We covered urinary system, so there's going to be anatomy there, but we also covered the chapter that was kind of a little bit of an extension of the concepts we've seen with the urinary system physiology, kind of extending it to the whole body level. So we discussed about water, electrolytes, and pH, and the balance of all of these three. So obviously, when it's such a physiological chapter, there's not going to be anatomy associated with it. So for this lab practical 3, focus on the respiratory system and urinary system, and you will be great. So what I'm going to do at this point, I'm going to stop sharing this screen and jump on to another screen that I think is going to be useful for us to spend a little bit of time. And that is, of course, the revision of the study guide. So like I said earlier on, we're going to start by talking about the respiratory system. And for some reason, my screen was working so well earlier, and now it's not allowing me to highlight. Let's give one more try. Yes, that's not doing what I wanted it to do. So let me just see. So in that case, what we're going to do is that we're simply going to work our way through the study guide, but we're not going to be able to do as much annotation as I wanted. I assume that it is probably due to the fact that we've got two things going on there with the sharing the screen and recording this session. So for that reason, we're not able to do the annotations on our study guide, but we are still perfectly capable of working our way through the study guide. And that's exactly what we shall do. So if you look at the study guide and you look at the emphasis on each part of the study guide, you'll notice that there's a lot of emphasis on respiratory system. Urinary system is equally covered, but when you look where the urinary system focuses, it's heavily on the nephron as a structure. It's heavily on the nephron physiology, things like that. So study guide is good in a sense that it kind of helps you to guide to know where to direct your attention. And finally, of course, the final portion of the study guide focuses on this fluid, electrolyte, and pH balance. So hopefully that will make you feel at ease having had a chance to see those topics. So we're just going to jump, in the interest of the time, right to the chapter on respiratory system. And as always, I try to start with kind of drawing these connections between different systems. So some of the things that we're going to see is that we relied on the cardiovascular system to assist us with the respiratory system. So for the transport of the gases that we're either getting into the body or we're removing from the body by using the respiratory system, that's where the cardiovascular system will assist by transporting these respiratory gases. We're also going to see that there's going to be pathogens that we have to protect ourselves from, also in the air. Recently, a lot of media attention has been on the COVID pandemic, and of course, that's an airborne pathogen. So we see very well why we need to have our immune system working with the respiratory system on that as well, protecting us against different kinds of pathogens. And finally, the final concept that kind of ties into what we've discussed, we are going to talk about the urinary system a little bit. And what we're going to see with the urinary system is that whatever we couldn't remove with the respiratory system, that's where the urinary system steps in, for example, helping us to get rid of some of the excess hydrogen. So that's one of the good examples where we do end up seeing the interaction with these systems. So if we try to really simply summarize why respiratory system is so important, it all comes back to this formula that we've seen before. We've seen that the formula of cellular respiration tells us how we are converting sugars. So in this case, I have a glucose molecule, and we're converting that to ATP. ATP was the cellular energy currency, so energy stored in a chemical format that can be then used to drive all other processes within the body. So for our body and for the cells in our body to be able to do this cellular respiration, there's two things that need to happen from the point of view of the respiratory system. We need to get oxygen to these cells that are doing cellular respiration. So that's where the oxygen intake comes in. And as a byproduct of cellular respiration, we're going to produce carbon dioxide. So we need to get rid of that somehow. And again, that's where the respiratory system is going to play a big role. So if you really wanted to summarize the two roles of the respiratory system as it relates to the gases, it's getting the oxygen into the body and it's removing the carbon dioxide from the body. So it's this interaction with the other body processes, supplying oxygen and removing carbon dioxide. That becomes a very, very important role of the respiratory system. In addition, of course, we're going to end up seeing that respiratory system does a whole bunch of other things as it relates to the speech production, but also to the sense of smell known as our olfaction. So there's a lot that respiratory system does. I'm just highlighting some of the key points. So having said all that, I think a good place for us to start this review of the respiratory system is by reminding ourselves of the different parts of this system. And let's do just that together. I'm going to just zoom in a little bit more. So we're going to see or start with a few words about the nose as the route of entry of the air to the body. We're going to talk about nasal cavity in a little bit more detail, why nasal cavity is so important, what functions it serves. We're going to have a look of those paranasal sinuses. You might remember them from when you covered your skeletal system and skeleton and especially the skull. So there were those empty spaces within the bones of the skull. Those were our paranasal sinuses. So we'll revisit that concept. We're also going to mention pharynx and larynx. Larynx especially, we're going to look at the cartilages that relate to that. We're going to think of the voice production as it starts at the larynx. And then we're going to continue further down the respiratory tree, respiratory tract, and talk about the trachea. In a layman's term, often referred as our windpipe. We'll see some changes happening within the trachea and then the branches that come off from trachea, the primary bronchi, secondary and tertiary bronchi, and then bronchioles. We'll notice that there's certain changes in the anatomical structure of this respiratory tree as we move downwards on it. And finally, we'll spend some time talking about the lungs and alveoli as well. When you're looking at these different parts of the respiratory system, one thing that you might notice very soon is that some textbooks divide it into upper and lower respiratory tract. And this transition point where the upper ends and lower starts varies a little bit depending on which textbook you're looking at, but for our purposes, if I were to ask you something in an exam about the respiratory system, how you divide it into upper and lower part, it would be something very obvious. So for the upper parts, the structures here higher on the list, lower parts, the structures here lower on the list. So I would not throw you a curveball. It would be something that would make sense and be quite obvious. So let's start with the nose. And a couple of things to notice about the nose when we talk about the respiratory system is that that's true, it's the structure through which we take in most of the air. There is an additional option of recruiting mouth for additional air supply to the body, but by default, we're really supposed to be nose breathers. And the reason why we're supposed to be nose breathers is that the nasal cavity serves so many purposes and is so important. However, we do end up seeing that we start breathing through the mouth when the needs are not met, when we're not transporting enough air in and out to the body through just nose. A good example of that I shared in the class was yawning. So if a student yawns in my class, I don't get upset and think that, oh, they're just bored. I think of the physiology behind there. So yawning is really your body's response to lowered oxygen levels. And what's a quicker, or is there a better or quicker way to get a lot of oxygen into the body than opening your mouth as wide as possible and taking that big yawn? Well, I can't think of a better and more efficient way. So for that reason, yawning really is just a way to respond to those reduced oxygen levels. Sometimes we see that we say that yawning spreads in a room, and I think that that's a good example of your physiological responses on that. With the nose, I wanted to also introduce the terms nares and nares as they refer to the nostrils. And within nostril, we also end up seeing some hair in there, especially as you age. I think older men have very pronounced sometimes hair within their nostrils. And if we think of the function of that hair, that's the first point now where we're starting to filter the air as it enters to the body. So remove any large particles before we let it further down the respiratory tract. So having said all that, I think we should shift our attention and talk a little bit about the nasal cavity and have a look what we can take away from that discussion. Well, nasal cavity doesn't have a skin layer inside. Instead, the layer is something that we know as mucous membrane. This mucous membrane has a thin skin, or it's not really skin, but thin layer. The layer is going to be sticky, and it's also going to be kind of warmer, humid surface. So all of this serves the three purposes that we have for the nasal cavity. It is sticky so that various particles get trapped into the mucous membrane, so we don't end up breathing in all that gunk that we shouldn't be breathing in all the way to the lungs. It's humid, so we can moisten the air before it enters to the lungs. Again, we want that to be humid so that the alveoli don't get damaged. And of course, there's a rich blood supply in the nasal cavity as well. A lot of surface capillaries. That's why you get a lot of nosebleeds kind of easily. They're there behind a thinner layer than what's skin in other parts of the body. So that allows us to heat up that air or warm up the air so that when it gets to the alveoli, it doesn't shock them. So all of this is possible thanks to goblet cells, which are these cells that produce the mucus of the mucous membrane. And from the mucous membrane lined nasal cavity, we're going to see that there's going to be three protrusions coming out from the lateral wall of the nasal cavity. And I've just quickly sketched, I had planned to sketch this on a computer as we talk, but since that's not happening, I'm just showing it here on the screen. I've quickly sketched the nasal conchae. They're simply just protrusions from the lateral wall of the nasal cavity, and these serve a two-folded purpose. First of all, these protrusions give us more surface area for this nasal mucosa lined nasal cavity. And another thing that it does is that these protrusions generate a flow of air described as a turbulence. So we're trying to make sure that as much of that air passing through these structures gets into contact with the nasal mucosa, so we can do all those three functions that we had a look of. The other 2 points that I have here listed that are going to be important are that the nasal septum, of course, referred to this structure separating the nasal cavity into right and left side, dividing the nasal cavity into middle. And we also saw that the floor of the nasal cavity was formed by a structure known as a palate. Well, what palate is, it's roof of the mouth or floor of the nasal cavity. And it can be divided into 2 parts. More anteriorly, and again, I just simply quickly sketched it for you. More anteriorly, we're going to have a hard palate. Hard palate is hard palate because there's actually a bony structure within that layer or within that structure. And more posteriorly, we're going to see soft palate. There's no bony structure, it's just softer tissues. The very end of the soft palate then turns into uvula, that dangly bit at the back of your mouth. And we're going to talk about the role of the uvula in a greater detail when we talk about digestive system. But at this point, those would be some of the good parts to know about the nasal cavity. Well, let's move on and have a look next of the paranasal sinuses. So you remember the term sinus always referred to some kind of an empty space or a cavity in the language of anatomy. And in this case, we're looking at the paranasal sinuses, so these empty spaces within the bony structures of the skull around the nose or around the nasal area. So those are what we're looking here. And this is where all that work that you put in to learn the bones of the skull will pay off, because these sinuses are named based on the bones where they are located. So we're going to have our frontal sinuses, one on each side, maxillary sinuses, again, one on each side. Sphenoid sinus is within the sphenoid bone. You remember that was the kind of a trickier bone of the skull, kind of much more three-dimensional, so further back in to the skull. Sphenoid bone is often described as a butterfly shaped, if you wish, so much more three-dimensional. And then we have our ethmoid sinuses. This is actually a group of empty spaces that are kind of interconnected. So I prefer to call it as an ethmoid labyrinth to really emphasize that kind of three-dimensional, multifaceted nature of the ethmoid labyrinth within the ethmoid bone. Well, then if we look at the roles and purposes of the paranasal sinuses, we're going to see 2 different explanations for why do we have paranasal sinuses. And again, some of these are better accepted, more well-established explanations, while others are more theoretical and still in the development. But regardless, we're going to go through those. So first of all, of course, if you compare why do birds fly and compare their skeleton to the human skeleton, one thing that you'll see very soon is that the birds have hollow bones. So we try to reduce the weight of their skeleton. So we're seeing a little bit of that same with the paranasal sinuses. So these empty spaces within the skull, we can consider that they help to reduce some of the weight of the skull, so that the muscles of your neck don't have that big of a job of holding it up. It's still a big job, but at least we reduce some of that weight load. We've done some experiments where we have actually prepared skulls that have both, just artificial skulls that have both paranasal sinuses and skulls without paranasal sinuses, and manipulated then the flow of the voice through these skulls. And what we'll end up seeing is that these paranasal sinuses make a significant contribution to the voice resonance that takes place in humans. So it does affect that as well. The third one that I have here, I think that that's more of a kind of a proposal level than a widely accepted. But you remember, similar as the cars had the front of the car designed that in an head-on collision, it on by design collapses, and with this collapsing kind of absorbs some of the forces, so we don't get as strong of an impact to the driver, so there's not whiplash as largely. So it has been speculated that paranasal sinuses could serve a similar purpose in case of humans. And of course, the final example of the functions of paranasal sinuses goes on with the sinusitis, the inflammation of sinuses. So remember, the term itis always referred into inflammation of something in medical terminology, and in this case it's going to be sinuses. So there is an immune system role potentially with the paranasal sinuses as well. So having said all that, one more structure that I do want to highlight is the opening of the auditory meatus at the nasal cavity. So this is a connection to the inner ear from your nasal cavity. And this connection is important because you remember the tympanic membrane separated the inner and outer ear. And for example, when you drive or take a plane and move on to higher elevation, you might have your ears popping. That's because the pressure changes in the outside air where we're at. Well, we need to balance of those pressure changes also with the pressure that's inside the ear. And one way to do that is to have the direct route of connection from the nasal cavity to the inner ear. So that's why kind of swallowing or chewing gum, things that make you kind of get air down in there, come in handy and quite useful if you're on a task to do that. So now that we've looked at all of these things, I think it's a good time for us to remind ourselves of the three main functions of the nasal cavity. Of course, it was to clear the air that's coming in. We want to remove any particles that would be an issue if we breathe them down into the respiratory tree and lungs. We also want to heat up the air that's going to go to the lungs and to the smallest structures, alveoli there. And we also want to humidify the air. So for that purposes, this nasal cavity had that mucous membrane lining sticky, kind of a wet surface, and we had a lot of capillary vessels within the nasal cavity to bring that heat with the blood to that area. So this way we make sure that the air that's coming in gets dealt in these three aspects. Another thing that we'll end up noticing is that if we want to make a comparison, what would happen if we don't do these, think of taking a deep breath of air on a cold winter's day when the air is really dry, and really taking that big breath into the lungs, you will probably end up coughing. That's because we haven't had a chance to do these three functions that the nasal cavity served such an important role to do. So I hope that that kind of helps to explain why aspect of this discussion. Next, I wanted to introduce the pharynx. So pharynx is kind of a skeletal muscle tube that runs from the back of the nose to the start of the larynx. And again, I'm going to sketch just a quick diagram to highlight few structures. And while I'm doing that, I just want to remind you that the pharynx was named based on the relation to the other locations that we're going to see. So the area immediately immediately behind the nasal cavity was known as our nasopharynx. The area immediately behind the oral cavity was known as our oropharynx. And the area above the larynx was known as our laryngopharynx. So pharynx is really divided into 3 parts based on the location of each of these parts. So I hope that that helps you as you're making sense of that structure. And that brings us to talk about the next structure, which is going to be the larynx. So let's have a look at the larynx. If you try to kind of position it, it runs somewhere between 3rd and 6th cervical vertebra, kind of starting from the hyoid bone. You remember hyoid bone was the kind of an odd bone floating on its own, held in place by connective tissues, muscles, and so on. Sometimes might get broken if someone strangled, so that might have some forensic relevance in that sense as well. But as we're talking about the larynx, one thing that I want to make very, very clear and emphasize is that even though we're going to be looking at some of the key cartilages that contribute to the structure of larynx, there's going to be a bunch of other structures, muscles, other elastic tissue that also contribute to the larynx. So do remember that they play an important role. And we're going to go through the different cartilages of the larynx next. So the largest one is going to be your thyroid cartilage. Thyroid gland is located just above it. Thyroid cartilage has one really prominent bump on it on anterior surface, and that's what you can see in a layman's language known as Adam's apple. So that's your thyroid cartilage. And again, you can kind of feel on your own body these cartilages, at least some of them. You remember Adam's apple was more prominent in men than women. Immediately underneath the thyroid cartilage is going to be our cricoid cartilage. Cricoid cartilage is the only fully fused ring in the respiratory tract. So all other parts are going to be open on the behind back of the larynx. Then we had the epiglottis, which was this flap-like structure that was kind of peeking a little bit out as well. And what we want to remember about the epiglottis was that it is open at all other times except when you're swallowing food or drink, when it comes down and closes the trachea, sealing it off, ensuring that the food goes down the esophagus rather than to the trachea. But at all other times it's open because ensuring that open respiratory pathway was a priority for us. Let's have a look at some of the smaller structures, and these following structures are going to be paired structures. So we're going to see 2 of these, one on left and one on right side. And we're going to start with Arithmeods. Arithmeods are these small kind of more of a triangular or pyramid-shaped structures at the back, kind of housed inside the thyroid cartilage space. There was a rich connection of multiple muscles there that helped to move them, and as they moved, they manipulated then the tension of the vocal cords. So those were our arithmeods. At the very top of the arithmeods, I'm just looking for my diagram, trying to locate it. At the very top of the arithmeods, we're going to see our cricoid cartilages. So cricoid cartilages are simply these little tips. I'm sorry, this is going to be our corniculate cartilages. So corniculate cartilages are these little tips on top of the arithmeoids. And finally, cuneiform cartilages are these cartilage parts housed within the mucous membrane lining of the larynx, not often seen very, very visible. So if you think of the functions of the larynx, we get to list few things that are important for us. First of all, of course, that's the route of air to the lungs. It also serves the purpose of separating the air pathway from where the food is going to go down the digestive tract, down the esophagus. So that's another important adaptation. And of course, this is where the vocal cords are housed. Couple of things about the vocal cords that we're going to end up seeing. Vocal cords, if you look at them in medical imaging or other ways, you'll end up seeing that they are kind of these paler structures. That's because there's no blood vessel supply on those, so they appear whitish in color. The space between the vocal cords is known as arglottis or sometimes remaglottis. And actually what's going to be interesting is that there's going to be two pairs of vocal cords. The more superior ones are known actually as our vestibular folds, and they are often referred as false vocal cords because they don't really contribute to the voice production, whereas the more inferior ones are the true vocal cords, known also as vocal ligaments, that contribute to the voice production. I do want to be very clear because I got a little bit of flashback, a slapback. Not flashback, a little bit of backlash back in the day for just leaving this story there. Do remember, of course, that the manipulation of the voice happens also further along at the level of the mouth and so on. So the next thing that I wanted to talk a little bit was to do with trachea. Trachea is about 4 inches long tube, very flexible. What you end up seeing with the trachea is that there's going to be these C-shaped rings, if you look from the top, that kind of keep the trachea open at all times. So even if you were unconscious, your trachea would still remain open thanks to these cartilage rings that ensure that this pathway remains open. If you look at a few other structures that we're going to see, when the trachea ends, at the very end of it, it divides into 2 primary bronchi. And this division point where the trachea ends and divides into 2 primary bronchi, it's known as carina. So it's going to be this kind of a tip or tent-shaped structure when you look with a camera down the trachea that's going to come visible there. So trachea divides into 2 primary bronchi. What we'll end up noticing also is that the our right primary bronchi comes down in a steeper angle than the left one. And for this reason, if we're looking for foreign objects, especially kids are really good, infants really good about this, inhaling in stuff that they shouldn't. If it goes to down the respiratory tract, we probably would find these foreign objects more commonly in the right lung because of this steeper angle. So that's something to keep on mind as well. The clinical example that I've given here is, of course, our tracheotomy, where we do an incision on the interior trachea if there's a blockage somewhere higher up to ensure that that airway is still open. So again, getting that air to the lungs remains important. If we keep working our way down the respiratory tree, another term that I wanted to mention was the term hilum. Hilum refers to this point where the primary bronchi penetrates into the lung for the first time. So that point is known as hilum. And then from the primary bronchi, we are going to have smaller and smaller and smaller branches, secondary and tertiary bronchi, bronchioles, terminal bronchioles, and eventually getting to the level of alveoli. One thing that we see as we move down this respiratory tree is that the amount of cartilage decreases as the amount of smooth muscle increases. So again, keeping this airway open, we're moving this task from the cartilage to the smooth muscle. A couple of structures, we mentioned a few of these already earlier on. I just want to remind you of those. The root of the lungs is going to be this vascular and bronchial attachments to the mediastinum costal surface really covers the outer exterior of the lungs against the ribs on anterior, lateral, and posterior sides. The apex of the lungs, remember that was the sharp tip in the heart. It was pointing inferiorly, anteriorly, and down and to the left. Whereas in case of the lungs, it's going to be this highest peak at the very top, the superior part, kind of going to the level of first rib through the level of clavicle. So lungs come up quite high up. That often surprises students. Base of the lungs rests on the diaphragm. So that large respiratory muscle, thin sheet of skeletal muscle, and then the hilum we already saw as the structure where the primary bronchi entered to the lung per se. Well, if we look at the lungs themselves, we'll see some differences between right and left side. The big difference is that the left lung is going to be smaller because much of the space in there is taken by where the heart would be. So this cardiac notch, this indentation is going to be visible on the left lung as well. What we're going to see is that since right lung is larger, there's going to be typically 3 lobes, superior, middle, and inferior one, separated by horizontal fissure separating the superior and middle one, and oblique fissure separating the middle and inferior lobe. On their left side, since it's smaller, we're going to typically see only superior, inferior lobe separated by oblique fissure. One thing to keep on mind is that we teach you averages, common things. It is not this black and white. So a lot of the times we do end up seeing that that there might be different number of lobes due to individual variation if we look at a larger population. But this is the average typical things that we see. Remember, lungs were enclosed within the pleural layers. So we've got our visceral layer and parietal layer, visceral being the innermost and parietal outermost. There's going to be fluid in between the two, helping to slide these layers past each other, add to that cushioning function, and most importantly, generate that negative pressure that keeps pulling the lungs expanded within the chest cavity. So that's why if there was a puncture to the chest cavity, we could have a lung collapsing. And if we look at the alveoli, this is of course the business end. Alveoli are these little microscopic sacs at the very end of the respiratory tract. And what we'll see with the alveoli is that these are so small that they easily get collapsed when we end up looking the microscopic structures. So really producing those microscopic slides is going to be a skill. They're small because that way we can pack a lot of surface area of the lungs into a smaller space. And this is where the exchange of the gases between the air that gets brought in to the alveoli and the blood vessels, the pulmonary capillaries, this is where it takes place. So this is the side where the gases get exchanged between the pulmonary capillaries. So having said all that, I think we're ready to move on to have a look of the physiology of the respiratory system. And we're going to remind ourselves of the 3 phases of ventilation. So first, pulmonary ventilation, referring to bringing the air from the outside world to the alveoli. Remember that breathing in required effort, so it's especially the diaphragm that's our main respiratory muscle that contributes to this. So try to take a breath in. That took effort, but if you just relax, air will automatically flow out. So expiration is going to be passive process, just due to the elastic recoil largely. You can, of course, breathe extra air out by forced expiration, which would be active process and require muscle contribution as well. Well, next, from the alveoli, we're going to see gas exchange. This is going to be external gas exchange, so that gives you a clue that we'll see the gas exchange again a little later. But external gas exchange refers to exchanging the gases from alveoli to capillaries and from capillaries to alveoli. And the driving force for this exchange of the gases was the partial pressure differences. So materials move from areas of high concentration to areas of low concentration. And then the gases get transported within the systemic circulation. And once we reach the level of tissues in the systemic circulation, it's going to be our internal gas exchange that helps oxygen to move away from the systemic capillaries to the tissues, and from the tissues we move carbon dioxide to the systemic capillaries, the blood within those. Remember though, even though this has been listed as 1, 2, 3, 4-stepped process, then it also works the other way around. We're taking gases away from the body. So that's something to keep on mind. There's going to be a bunch of non-respiratory air movements also that you might want to be aware of. So a lot of other important stuff such as coughing, sneezing, crying, laughing, hiccups, and yawning that we spoke about earlier on. Well, having said all that, there's two terms that I want to emphasize. Remember the term surfactant. This was a lipid protein complex that did two-folded thing. First of all, it helps to reduce the surface tension at the level of alveoli, so this exchange of the gases occurs more efficiently, but it also prevents these alveoli, these very, very thin, small microscopic structures, from collapsing. Term compliance instead, I think of it as referring to the measure of the stretch that the lungs have. You've got a little bit more complete description there at the top as well. But I want to move on to talk a little bit about spirometry, which is measuring these air movements in and out of the lungs respiratory system that can give us a good clue about patients' health in terms of the respiratory system. So let's start with volumes. So the amount of air moving in and out in normal breathing is not huge compared to the total capacity that we have. This amount of air moving in and out in normal breathing is known as our tidal volume, often abbreviated as TV. You can, of course, breathe more air into the lungs. This would be our inspiratory reserve volume, IRV. So how much more air you can breathe in, that's known as our IRV. And you can do the opposite. You can breathe extra air out of the lungs as well, and this would be known as our expiratory reserve volume, ER. Of course, there's always going to be some air stuck that doesn't get breathed out, some volume in the lungs and respiratory tract. This is known as our residual volume, so RV. Some air that always remains in the lung and respiratory tract. If we take our respiratory volumes, we can use them to calculate respiratory capacities. Let's have a look at that together. So if we're talking about our inspiratory capacity, IC, that refers to the volumes of adding the tidal volume, so the normal breathing volume, and the inspiratory reserve volume together. So that's how much we can breathe in, both normally and the extra that we can do. If we talk about our functional residual volume, well, functional residual volume gets calculated by adding together our RV, which was our residual volume. You remember that air that never left or goes anywhere, and our expiratory reserve volume, ERV. So the amount of air that can be forcefully breathed out. If you add the RV and ERV, that gives you the functional residual capacity. The next measure is going to be our vital capacity. Vital capacity is calculated by adding together the tidal volume, normal breathing rate, IRV, the extra that we can breathe in, and ERV, the extra that we can breathe out. So TV, IRV, and ERV added together gives us our vital capacity. And of course, if we want to make sense of the total lung capacity, we just add all of the three volumes together, our tidal volume, our inspiratory reserve volume, our expiratory reserve volume, and our residual volume. So that gives us our total lung capacity. And like I said, when we talked about the residual volume, there's always going to be some air stuck, whether it's due to some parts of the lungs not working that well, or whether it's due to the respiratory tract, just we can't fully empty it. These are known as our dead space. So you can add both together to get the total dead space of the respiratory system. Next, I wanted to say a few words about the gas exchange. We saw the external respiration between the alveoli and pulmonary capillaries, and internal respiration between systemic capillaries and the tissues. And I don't expect you to memorize these numbers, but what you'll notice is that the gases move from areas of high concentration to areas of low concentration. So it was the process that was driven by the partial pressure differences. One question that I do like to ask in an exam is that how do we move these gases in the blood then? And of course you remember that most of the oxygen was transported bound to hemoglobin, about 98%, whereas with the carbon dioxide there's more going on. So about 3/4 of carbon dioxide gets transported in blood as bicarbonate. Remember the carbonic acid equation that we had a look of multiple times. About 15% bound to hemoglobin and rest 10% dissolved into plasma. And final concept that I have here is a reminder of the hyperventilation. So hyperventilation referred to when we were losing too much carbon dioxide, and that caused us issues that we needed to pay attention. And at this point, what I'm going to do, I'm going to take a quick break because I do have another meeting that I have to jump into, but hopefully we're going to be able to come back to this and finish off this recording so that you have a complete review of the study guide. If not, I will upload a second video, but let's try if we can take a pause and come back in just a minute. Bear with me. Okay, and now that we're back, what I think we're going to do, we're going to finish off our review of the study guide. So let me share my screen once more. And I think this is where we finished just before I had to take a quick break. We basically discussed all that I had on mind on the study guide regarding the respiratory system. And what I'd like to do next is quickly revisit the urinary system and some of the key points from that as it comes to what you want to know for preparation for the exam. So urinary system and kidneys have a lot of functions. There's a lot more. I just listed some that obviously stood out for me. In the past, when we started this course and we talked about the blood and cardiovascular system, one of the big functions that related to the kidneys that we saw was to do with regulating the blood pressure. But there's also going to be things such as removing materials, removing the excess hydrogen as we see, regulating the fluid balance, and so on and so on. So there's a lot that goes under the kidneys. But what I really wanted to start with was to kind of review a little bit the external anatomy of the kidneys, that what's important, what do we know about kidneys, what do they appear, and then have a look of some of the internal structures as well. So we're going to have two kidneys, most of us, one on the left, one on right side. They're kind of roughly bean shaped, but obviously larger in size. And an interesting thing with the kidneys is that they're going to be, if you compare or look at them in the body, almost like a mirror image of each other. So the area that's facing to the middle, the medial side, and I'm just trying to make sure that I'm showing a diagram. Medial side is always concaved, whereas the lateral side is gonna be then our convex sides. So we're gonna see the same arrangement regardless on which side kidney we're looking at. Within that medial side, the side that's our concaved side, there's going to be few structures that are we're wanting to be familiar with, and the renal hilum refers to this area where the ureter comes out and blood vessels come in and exit. There's going to be nerves, there's going to be lymphatic vessels, all that good stuff entering and leaving the kidney. And of course, the lateral sides, like I mentioned, were more convex than in their shape. Kidneys, often when we dissect them, appear kind of shiny, smooth surface. That's because they're enclosed inside a renal capsule. Kidneys are also quite far back to the back, so many students are sometimes surprised about that. And we will end up noticing that there's a slight height difference on the left and right side when we look at where the kidneys are located. And this is with the right side kidney being a little bit lower than the left side. And this is because immediately above the kidneys, we're going to have our liver. And on the right-hand side, that liver takes a little bit more of the space. So that's why the right kidney is lower than the left. So that liver kind of explains that height difference that we're seeing. If we're looking at the internal anatomy of the kidneys, we're going to see a few things. And I'm just going to sketch really quickly, just kind of an enlarged diagram of a kidney, and we're gonna see, just like we had with the brain, you remember that brain had the cortex, which was the outermost layer. Similarly, the kidney will have a cortex, which is the outermost layer. So this is the outermost layer of the inner structures of the kidney, and the other material within the kidney is then going to be the deeper structure. It's going to be our medulla. So those two parts I want to separate. Within the medulla, we're going to see these kind of pyramid-shaped structures, if you wish, or more triangular structures, however you wish to describe them. And these are often darker in color, with the tissue colors when we're dissecting into the kidney. And these are what we know as our renal pyramids. So these darker medullary structures within the kidney. And the space between the renal pyramids then, which is going to appear paler in color, these are going to be known as our renal columns. Of course, the part where we get the final product from the kidney slowly collected through calyxes and being carried then towards our ureter. That's gonna be that region, our renal pelvis. I've listed the flow of the production of the materials there as well for your handout in case you find that useful. The next structure that I have here, I wanted to talk a little bit about nephron. And the reason why we're talking about nephron is that it is really the structural and functional unit of the urinary system and the kidney. It's the business end. I've listed the parts of the nephron here for you. I didn't mark them on this diagram, but we can go through them together. So remember, this is a simplistic, simplified diagram showing the general structure of a nephron. So we've got our afferent arteriole coming in and efferent arteriole leaving. So that's the blood supply to the kidney. Between afferent and efferent arteriole, we've got a capillary tuft, which is known as our glomerulus. Remember, this picture is almost like a cross-section of a nephron. So the capillary tuft would be here, and around it, there's going to be the first actual part of the proper nephron, which is going to be our Bowman's capsule or glomerular capsule. And it is within here that we filter materials away from the blood and it enters to the nephron. From the glomerular capsule, we're going to have a proximal convoluted tubule and then our nephron loop, or sometimes also known as a loop of Henle, and then distal convoluted tubule. And distal convoluted tubule eventually opens up as a collecting duct. So let's have a look of, now that we've looked at the anatomy, let's have a look of a little bit more of the physiology, movement of the materials that we end up seeing here within the nephron. And we're going to start with this where I already referred that from the capillaries of the blood vessels, material moves away into the nephron to the glomerular capsule. Sometimes glomerular capsule is also known as a Bowman's capsule. So this process of removing materials from the blood to the nephron is known as our filtration. On the more proximal part of our nephron loop or loop of Henle, another movement takes place where materials leave the nephron back to the vasculature. This is known as our reabsorption. We will see an opposite process of this taking place at the distal nephron loop. From the blood, material moves back into the nephron. This is known as our secretion. And finally, the final product, the urine that's being produced, that leaves the distal convoluted tubule and enters to the collecting duct, that's going to be our urine, and that's known as excretion. So I hope that those movements make sense. Think of it as a kind of an designed structure. So we have multiple chances of fixing if something moves the way where it shouldn't. So we kind of have a over-engineered urinary system. That's why we have so little urinary system issues with the aging. It's designed to facilitate those changes and misfunctions that might take place. Couple of terms that I wanted to mention. First of all, I wanted to mention a term, glomerular filtration rate. Glomerular filtration rate is a measure that we use a lot in clinical practice to tell us how well your kidneys work. So that's a measure for how much blood passes through the glomeruli in 1 minute. So we're looking at really how well your kidneys are working. If you have a high GFR number, we know they're working well. If GFR number is too low, that will then cause issues that we cannot use certain medications or contrast agents in medical imaging and so on. Another one that I wanted to highlight is our hormone that we're familiar from the chapter on endocrine system, ADH. ADH stands for Antidiuretic Hormone. And what Antidiuretic Hormone does, it does what it says in the tin, makes you urinate less. And how it does that is that it turns the distal convoluted tubule wall more permeable so that now materials leave the nephron back to the vasculature at the distal convoluted tubule. So now we're starting to see explanation to how these hormones acted, whereas before I've just always told you, just trust me, that's what they do, but I've not provided you an explanation. So that's how ADH worked. Let's finish off the urinary system by looking at the other structures. So ureters are these smooth muscle tubes that carry the urine from the kidneys to the urinary bladder. Because they're smooth muscle tubes, they can do this kind of movement of moving the urine along that. So it's not just the gravity that moves urine from kidneys to the bladder. This movement would actually happen even against the gravity, thanks to this smooth muscle lining of the ureters. Next, We're going to see urinary bladder. So urinary bladder is a muscular pouch. That's the temporary storage for urine. So until we're ready to relieve and empty the urinary bladder, we're able to keep it in there, located at the pelvic floor behind the peritoneum. You'll remember that the ureters entered to the urinary bladder from the bottom part of it. And there's going to be two openings of the ureters, and they're more posterior if you see this sketch that I just prepared there. And there's going to be one opening for the urethra, which is, of course, the path for the urine to the outside world. So in total, at the bottom of the urinary bladder, we're going to see three different openings. And if you have a little bit of imagination, you can almost see like these three openings would form a triangular shape or structure. Obviously, there's not going to be a real triangle there in the real urinary bladder, but keep that as a thought. We call that triangular structure as a trigon. So it's an important anatomical landmark that we might use to identify this area of the urinary bladder. Finally, I wanted to talk a little bit about urethra. Urethra is the tube that carries the urine out from the urinary bladder to the external world. There's going to be two sets of sphincters. We're going to see internal and external urethra sphincter, and we saw big difference between sexes here. Male urethra is longer than the female one, and this results in females being more prone to UTIs, urinary tract infections, as it is shorter journey for pathogens to travel up that bit and cause issues. The term that we're going to use when we talk about urination in proper medical language is micturition. So you can use that term next time rather than say, I need to go to loo. You can say that I need to go and practice some micturition. In terms of the urine composition, obviously most of it is water, and then we're going to have various different solutes and other materials that we find in the urine. The things that we're looking is the amount of material too high or too low? We're also looking at, are we seeing something that shouldn't be there? So blood proteins, white blood cells, bile pigments, things like that. So there should not be blood in the urine. If there is, that's a sign that there's some issue either with the filtering at the glomerulus, or there could be damage somewhere along the urinary tract. White blood cells would be an indication of some kind of an infection, and so on. So we're checking for those. And that really is all that I wanted to say about the urinary system, which brings us to the last chapter that we've yet to review, which is going to be our fluid, electrolyte, and pH balance. And of course, this is important so we can understand better the test results that you're going to see. I do always ask about the water content amongst different groups in the exam. And what we'll end up seeing is that the infants have higher body water percentage than elderly people. And this can be explained with the fact that infants have a kind of a difference with the elderly people in their body fat percentage, bone mass, and so on. So infants have somewhere in the range of 73% of their body weight is water, whereas in elderly it can be as low as 45%. We'll also see that with the adults, there's a sex difference, with males having generally a little bit more water content than adult females. This can be explained with the, again, body fat percentage and skeletal muscle mass and so on. So I tend to ask this in an exam, can you organize these into a correct order? So that might be a fun thing to know. If we think, where is this water then in the body, we'll end up seeing that about 2/3 of all the water in the body is inside the cells, whereas the remaining third is really covering three different groups. It covers the plasma of the blood, the interstitial fluid, the fluid between the cells, and also the lymph. So we talk about intracellular fluids if it's inside the cells and extracellular fluid outside the cells. We have already seen the characteristics of water that make it so useful for sustaining life. So this is probably largely a revision. You remember that water was great transport media carrying a lot of things such as most of the blood plasma being water, but also carrying nutrients into the plants with the water and so on. The concepts of cohesion and adhesion, let's have a look at those. So cohesion refers to the attraction of water molecules to other water molecules, whereas adhesion refers to the attraction of water molecules to other polar surfaces. So things like positively or negatively charged surfaces. Well, surface tension, which we sometimes see examples as, for example, bugs walking on water, is a direct consequence of the cohesion of the water molecules being attracted to other water molecules. We also saw how water was great in binding heat and releasing heat slowly. So we saw this in this kind of lesser temperature changes on the coastal area. But think of it also in a human body as a way of transporting heat from one body part to another. For example, from the core of the body where there's more heat during a cold winter's day when you're outside, your nose tips, ears, fingertips might be getting cold. So we're transporting that heat from the warm parts with the blood to these parts that would otherwise get a frostbite. Of course, water can take three different phases: solid, liquid, and gas. Plasma is not one of the phases of the water, but I added it there because I wanted to discuss about that in a class, so we're not going to go there now, but you know that some materials can also take plasma phase. What binds the transport media and great solvent points together when we're discussing of the water is that water, other than it's a great for carrying things, it's also a great solvent. So most things dissolve into water. And there are materials that don't dissolve completely, such as glucose, lipids, ureas, creatine, but also some that do. And as they dissolve into the water, there's a change in the charges. So we're forming either positively or negatively charged materials. If nothing else, the big take-home message from this portion was that remember that water tends to follow electrolytes. So wherever electrolytes go, water tends to follow, and the other way around, that wherever water tends to go, electrolytes tend to follow water then. So these two are going to be interdependent, dependent on each other. And I tend to ask that question that what's the relationship between water and electrolytes in my exams every time. So that's a good one to know and feel comfortable with. And let's continue our journey revising these key concepts from this chapter. We, of course, saw if the intake of water and loss of water are in balance. We talk about body being in homeostasis. So you should roughly take as much fluids as you're losing. Main method of getting fluids to the body, about 2/3 of all the water intake, is through drinking. So thirst is the driving mechanism. Also through moist foods, about 10% as a byproduct of metabolic processes. We lose most of the fluids from our body through urination. Also evaporation, sweating, and breathing plays important roles as well. I discussed during the class the osmoreceptor ADH mechanism using the homeostasis as a way to explain it. So remember, homeostasis always referred to the maintenance of consistent internal environment, and I provided the little diagram here showing the steps: stimulus, some sort of a change, we detect it, pass it to the control center, which is typically brain, compare it to the normal values, and if necessary, we act to stop the stimulus from happening. That would be negative feedback. And we can apply that to explain two conditions: when we have too much water in the body versus when we have too little water in the body. And these were covered earlier on, so I just kind of wanted to offer you a little bit of a diagram there that you can refer back to if you wanted to remind yourself of those concepts. Finally, I wanted to talk a little bit about the tonicity. So you remember tonicity was important, for example, when we talk about the electrolytes and water following them. So a quick review, what we've discussed, hypotonic conditions. So hypotonic cells were those hippo cells. That was my mnemonic. So big swollen cells. So water is flowing into a hypotonic cell, and that causes this cell to grow, swell, and it might eventually even burst. Isotonic cells, you remember my mnemonic for that was isoperfect. These cells gain some water, lose some water, but overall there's no net change in the water content, and the cell remains the same size. It's perfect. It doesn't need to change. Finally, hypertonic cells. Hyper cells in my mnemonic were the skinny cells. So in this case, water is flowing outside from the cell, and the cell is shrinking, kind of drying up. So that's something to keep in mind. This is relevant for us when we talk about, for example, recovery drinks. If a person loses a lot of electrolytes, we want to replenish those, because otherwise we might have issues with tonicity, that water is going where it shouldn't be going, and we'll end up in trouble. We also talked a little bit about the pH scale, noticing that the H letter there stood for hydrogen. So pH scale tells the concentration of hydrogen ions, ranging from 0 to 14, kind of middle range, around a middle number around 7. And you can be either too alkalinic or too basic or too acidic. What we noticed when we talked about this is that your body does an exceptional job of keeping the body pH within such a narrow range, usually within 0.1 units, whereas the survival range even is still narrow, from 6.8 to 8. If it's any higher or lower than that, we end up with issues with alkalosis or acidosis. Well, how is your body able to do this, maintaining the body pH within this narrow range? That's thanks to 3 different mechanisms that play a role. First of all, buffers are chemicals that bind into acids or bases, bringing the pH close to neutral, act very fast, very immediate effects. Respiratory response takes minutes, so we remove materials through the breathing out. And finally, we discussed about the kidney mechanism, renal mechanism, which takes longer time, from hours even to days, to remove via kidneys in urine materials that might affect the pH. This is most efficient, but also the slowest method. So the effects are long lasting, very, very large, potent changes. So it's usually the interaction of balance of these 3 responses that allows us to keep the body pH within this narrow range. And like I mentioned, if our body pH is not where it needs to be, we can end up with medical conditions. Final thing that we had a look up was that when you went to doctors, and often you end up getting an IV drip. This drip contains sterile water that contains just the right mixture of those electrolytes, so we don't end up with tonicity issues. It's also a great way for us to get quickly medicine into your body through an IV. So that's why we like to put that as well. And having said all that, I really think that that completes our review of the study guide for this chapter. I know that it's been a lot that we've had a look of, and I'm, like always, uploading this recording also to your Canvas. So in case you missed the review session earlier this week in person, you have this to come back to. But remember, I don't upload the completed study guide anywhere, so this is really your chance to kind of get that information if you were not able to come, but you have to watch through that video. I think in this evening's review session, we'll have a look of the exam and practical a little more. There is a good chance I'm expecting few students to join as well. So if they have other questions, then we'll just adjust the content of the evening's review session to match with that. But that's the initial plan. I hope that that works for you. And as always, do not hesitate to reach out to me. I'm here every step of the way to make sure that you reach those goals of yours. and you're gonna ace this course and get great grades. Remember, we're 3/4 in now, so let's finish off strong the last final quarter of the course and get you those excellent grades that you deserve. And having said all that, I'm gonna wish you very good rest of the day, and some of you I might see tonight. Thank you for joining me. Have a good day. Bye.